Hello, my dear students. I'm Ifat, and today I'm going to discuss with you about history of microbiology. Last classes we read about what is microbiology, why we study microbiology. With that, we also read about classification of Aristotle, classification of Carolus Linnaeus with different living organism and microbial world classification. With it, we also learned about different characteristics of the microorganism. Today we'll observe microbiology with some experiment of history. Okay. So first of all, how the cell was invented. The cell was invented actually by Robert Hooke in 1665. He actually observed some corks of bottle and he showed that there were some little boxes there and he named it as cell. But in this way, cell was invented. After Robert Hooke, another renowned scientist who known as Antony von Leeuwenhoek, actually he was first person who invented microscope and discovered microbial world. Okay, and Leeuwenhoek microscopes actually magnify the objects more than 50 to 300 times than normal is size. And he actually prepared that microscope with double convex glass lenses which he held between two silver plates, and this is actually his invented microscope. Okay, and with his invented microscope he observed a variety of things. What was that thing? That is rainwater, pond water, blood, even some darts which is known as scraping of his own teeth. And whenever he just magnifying that different kinds of materials or different kinds of things, he saw that some little animal cubes which he named, actually there were some moving objects which just moving around, and he was so much surprised that he just sketches uh, that animal cubes and communicate with his finding with the Royal Society of London, who at that time it was a famous actual organization who just published different kinds, of articles, okay, renowned articles. So he just contacted with Royal Society of London and he just published his first article in Philosophical Transaction, which was Royal Society's journal. And after his publication, actually, the microbiological things and with the different kinds of experiments that related with microbes, actually, it's go faster. Okay, and at that time he just gave one theory or discovered one theory that is called abiogenesis or spontaneous generation. It means that any kind of non-living things can be produced or can born from non-living material. Okay, so it's called actually spontaneous generation or abiogenesis, and it's like that every living materials can born from any non-living materials. But after his theory, after giving this theory, different scientists, physicians, doctors worked on it for proving this theory and non-proving or disproved of this theory. So one of them was Francesco Redi. He actually believed, he was a physician, and he actually believed that it's not possible to born any kinds of living materials from any non-living materials. So he just showed an experiment. His experiment was, there were three jar. One is all of the jar contained the meat broth, and one jar head was open, one was just covered with a core, and one was covered with a net. Okay, and after some days he just checked that the two jar that is covered with core and covered with net, there is no growth of any kinds of larva of flies. Okay, and it is called maggots. Actually there is no this growth of that. But another thing, another jar that was actually open headed, there different kinds of larva just he watched. So from that tree, he wanted to say that actually these larvae just came from with the help of these flies, but he couldn't prove that. Okay, he was not so much strong proof on it that these flies actually carry that larva, okay, or carry that microorganism. So he just gave that experiment, and others actually worked on his experiment again for disproving his theory that actually living materials cannot grow from living materials. So another scientist who was John Needham, who actually strong supporter of the spontaneous generation theory. That means any living materials can grow from non-living materials. And for that reason, he proposed that any kind of animalcules or tiny organism can grow from in the mutton gravy automatically from the jar, or from the mutton gravy that was grow actually. And for that reason, he covered the flask with cork as done by Reddy. And that means he covered these flasks also with the cork. So three flasks actually covered with the cork on neck. And after some days, he showed that automatically here the larvae born. So he strongly said that any kinds of living organism can be grow from non-living materials. That means that grow from that jar or that growth. So after his experiment, another scientist who was known as a priest actually, that is his name was Lazarus Plezeni. He actually again worked on Reddy's work or Reddy's experiment, and for that reason he boiled that beef broth for an hour and sealed the flux for a long time, and he then observed that there is no appearance of microorganisms. So he first disproved the theory of spontaneous generation and proposed the theory of biogenesis. Okay, that means it is called the theory of spontaneous generation is also called theory of abiogenesis. He disproved that and proposed the theory of biogenesis, and he said that every form of life take its origin from their parent germ cell or cells. That's without parent cell, it's not possible to grow any kinds of microorganisms, but he cannot give any experiment on it. He just give the theory. Okay, so we just get theory from him, not any experiment. So there were some confusion on it, that is it possible or not. After Lazaro, we Pasteur proved that theory, and he proved the theory of biogenesis, and this proved the theory of spontaneous generation. And after his experiment, actually, the theory of abiogenesis or spontaneous generation actually totally cancelled, and it's now established the theory of biogenesis. And he also called father of microbiology. Why? Because it's lots of contribution to the microbial world. So the experiment was like that. He taken that one plug and he poured some non-sterile liquid there. That is the non-sterile liquid. And after that, he added another swan neck with that flask, and he just sterile that flask. Okay, that is the sterilization of that flask. After that sterilization of that flask, he just sterile the liquid within the flask, and he just keep that flux like that. After cooling that, after a long time, he just observed there is no growth of microorganism here. That is after a long time, and his cork was open-ended, so lots of dust actually just keep there or deposited there. Okay, and after some days, suddenly the flask actually tipped, or you can also say it moved, and due to moving, the dust that is deposited from long time, it just go within the sterile liquid, and after, within short time, he observed that the whole liquid actually spoiled and microorganism grow. And from this, he just strongly said that the theory of biogenesis actually the right. For growing of any microorganism, its parent cell is very much important. Without parent cell, it's not possible to grow any microorganism. Okay, So that was actually Pasteur's experiment on swarm of plugs, which proved the theory of biogenesis. With that, it is actually explanation, nothing else that I just say. And with that, he also give different kinds of experiment, different kinds of theory. With that, most common is fermentation, that pasteurization is given by him. And he also founded another theory that is called germ theories disease. And this theory, he just given or proposed that theory, but he cannot give he couldn't give any kinds of experiment on it. Okay, so this theory actually like that, for growing any kinds of disease, any fixed microorganism needed. Without the any one microorganism, disease cannot actually born in our body. That means any one kinds of microorganism can produce disease in different kinds of animals or different kinds of human beings. But as there was no experiment, or there is a proved experiment on it, he just known as only the founder of the thesis in this theory actually, or this germ theory of disease. And on this germ theory of disease, then two scientists actually worked on it. And these two scientists was, one was Robert Koch and another was his teacher, that is Henley. Okay, so both of the scientists actually proved that germ theory of disease. And how they prove it? Actually, they taken a dead lamb which died due to some specific reason or due to some specific microorganism. Yeah, they just took some blood from that dead lamb or diseased lamb, and he cultured that and prepared a culture media of that microorganism. And then he taken this microorganism and pushed into a healthy rat. And after some days, they observed that the rat is died, and they again take the blood from that rat, rat, the died rat, and they again prepare the culture. And then they just compare between two cultures, and they observe that the both of the culture are same. And from that, they just prove that any microorganism can create disease in different kinds of animals. Okay, and the microorganism was antibiotic. So we learn about some experiment, and we also learn about some scientists. With that, he also called the father of the Robert Koch, known as father of bacteriology or practical bacteriology, as he actually invented bacterial films, glass slide, different kinds of needles, and he also discovered the tubercle bacillus or this microorganism. So he also had lots of contribution to the microbiology. So he is known as father of bacterial uh, practical fathers of practical bacteriology actually. So with there, these are the actually some activities of scientists that are very much famous in microbiology. With this experiment, these are some scientists who also has different kinds of contribution to the microbial world. As Alexander Fleming, he was very popular and he discovered the penicillium from the Penicillium notatum. Like that, these experiments and these scientists name and all of the disease and microbes name is for all of your learning. So just note down your problems, which we will discuss in our tomorrow's class. Thank you for today. Allah Hafiz.